Welcome to this lecture on one of the most fascinating and notorious creatures in the field of parasitology, the Candiru. Today, we are going to explore how this tiny Amazonian fish survives, its unique biological mechanics, and the surprising mathematics behind how it hunts its hosts in pitch-black waters. By the end of this session, you will understand both the biology and the physics of its survival. Let us dive in. Now that we know our topic, let us start with an intuitive picture of how this creature operates. Imagine a heat-seeking missile, but instead of tracking the heat of an engine, it tracks the faint, invisible smell of chemicals. The Amazon River is incredibly muddy, meaning visual hunting is nearly impossible for the creatures that live there. The Candiru is a tiny, almost transparent fish that acts very much like an aquatic vampire. It does not hunt to eat solid meat. Rather, it hunts exclusively to drink the blood of other living creatures. It waits patiently in the dark water for a larger fish to exhale ammonia from its gills, and then it follows that invisible chemical trail right to the source. Building on that intuitive picture, let us look at the formal biological definition. The Candiru is scientifically known as Vandellia cirrhosa. It is a species of parasitic freshwater catfish native to the Amazon basin. What makes it truly special is that it is one of the very few known vertebrates that is an obligate parasite. This means it absolutely must feed on the blood of other animals to survive and complete its life cycle. Morphologically, it is perfectly adapted for this stealthy lifestyle. It is small, slender, and translucent, making it incredibly difficult for potential hosts to detect its approach in the murky waters. Understanding its shape leads us directly to its most crucial anatomical feature. The Candiru is equipped with specialized backward-pointing spines located on its gill covers, which biologists call the opercle. These spines are biologically crucial for its survival. Because the Candiru feeds in fast-moving river currents, it needs a way to stay attached to its host. Once the Candiru enters a tight space, such as the gill cavity of a larger host fish, it flares these spines outward. This mechanism locks the parasite permanently in place, ensuring it is not washed away while it feeds on the host's blood. This highly specialized anchoring mechanism unfortunately brings us to its notorious reputation in human medicine. While its natural hosts are other fish, the Candiru is infamous in medical parasitology for accidental human parasitism. It allegedly enters the human urethra by mistaking the urea in human urine for the ammonia excreted by fish gills. Once it follows this chemical trail and anchors inside the human host using those opercular spines, it uses its needle-like teeth to bite into an artery. At this point, it does not even need to suck the blood. It simply allows the host's own blood pressure to pump blood directly into its stomach. Beyond the shock value of human infection, why do scientists study this creature? Studying the Candiru is absolutely vital for understanding the evolution of parasitism in vertebrates. This is a phenomenon which is incredibly rare when you compare it to invertebrate parasites like ticks or tapeworms. Furthermore, it provides a unique real-world case study in chemotaxis. Chemotaxis is the biological process of how organisms use chemical gradients to navigate their environment. By studying how the Candiru detects ammonia, biologists gain deep insights into sensory evolution and how animals adapt to environments where vision is completely useless. This evolutionary marvel does not just interest biologists, It has direct applications in modern technology. In the fields of medical parasitology and tropical medicine, understanding this parasite is obviously important for treating and preventing those accidental human infections in the Amazon region. But even more fascinating is how the Candiru has inspired human engineering. The biomechanics of its backward-pointing opercular spines have inspired modern engineering designs for anchoring mechanisms in soft tissues. Today, the exact same mechanical principles used by the Candiru to lock into a fish gill are being used to design life-saving medical stents and surgical staples used in human hospitals. Now that we understand the biological and engineering significance, let us look at how the Candiru actually finds its target. To truly understand how the Candiru finds its host in pitch-black water, we must look at the mathematics of chemical diffusion. When a host fish exhales ammonia or a human excretes urea, that chemical spreads out into the water, becoming less concentrated the further away it gets. We can model this chemical tracking mechanism using a standard exponential decay formula for concentration gradients. This mathematical model helps us predict exactly how close a Candiru needs to be before it can sense a potential host. This brings us to the specific mathematical formula that governs this biological detection system. The formula is C of x equals C naught times e to the power of negative kx. Let us break down what every part means. C of x represents the concentration of the chemical at a specific distance from the host. C naught is the initial maximum concentration, right at the source, like the host's gills. The symbol K represents the diffusion decay constant, which dictates how quickly the chemical disperses and depends heavily on water currents. Finally, X is the physical distance from the host in meters. Physically, if the distance X increases, the concentration decreases very rapidly, meaning the Candiru must be within a specific range to detect the chemical threshold. Before we move on, let us see this formula in action with a concrete example. Suppose a host fish exhales ammonia with an initial concentration of 100 units. The decay constant in calm water is 0.5 per meter, and the Candiru's minimum detection threshold is 10 units. We need to find the maximum distance x where the concentration equals 10. We start by substituting our known values into the equation. Next, we divide both sides by 100 to isolate the exponential term, giving us 0.1. To remove the exponential function, we take the natural logarithm of both sides, which gives us approximately negative 2.30. Finally, we divide by negative 0.5. The negative signs cancel out, leaving a positive distance of 4.6 meters. This means the Candiru can successfully detect the host from up to 4.6 meters away. Now that we have calculated distance, let us try a different scenario involving strong river currents. A human is urinating in the river, creating an initial urea concentration of 500 units. A strong current increases the decay constant to 1.2 per meter. If a Candiru is exactly 3 meters away, will it detect the human? Its threshold is still 10 units. We plug our values into the right side of the equation. We multiply the decay constant by the distance to get negative 3.6 in the exponent. Evaluating the exponential term gives us a very small decimal, 0.0273, meaning only about 2.7% of the chemical remains. Multiplying this by the initial 500 units gives a final concentration of 13.65 units. Since 13.65 is greater than 10, the Candiru will successfully detect the human despite the strong current. To fully grasp what we just calculated, let us look at this visual representation of the chemotaxis gradient. On the left, you see the host fish acting as the source of the chemical gradient, represented by the blue radiating waves. Notice how the color fades as it moves outward, illustrating the drop in concentration. Along the bottom, we have the physical distance measured in meters. Above the fish, there is a graph plotting the exact mathematical relationship we just used. The vertical axis is the chemical concentration, and the horizontal axis is distance. The curve clearly shows the exponential decay of the concentration. The Candiru, shown on the right, must swim within the specific threshold region under this curve to successfully detect the host against the opposing river flow. We have covered a lot of ground today, so let us summarize the key takeaways. First, the Candiru is a rare example of an obligate vertebrate parasite, perfectly adapted to the dark waters of the Amazon. Second, its specialized backward-pointing opercular spines allow it to securely anchor inside host cavities, a mechanism that has even inspired modern medical engineering. Finally, because visual hunting is impossible, it relies entirely on chemotaxis to locate hosts. As we proved with our calculations, this chemical tracking is governed by the strict mathematics of exponential decay. You now understand both the fascinating biology and the underlying physics of this incredible parasite.